Good morning and welcome this morning to a webinar that I'm going to take you through today around turning molecules into medicines. My name's Gail Marshall and I'm a lead scientist at the Medicines Discovery Catapult, and my focus is around biomarkers. And what I want to share with you today is what drug discovery translational science research can do, and then how the Catapult can certainly help you with the challenges that faces. So here we go. Okay, so there's been quite a lot of research over the last few years around why drugs fail in the clinic. So most of these researchers come back with it's more than 15 years and around 2 billion per medicine that's been discovered. And whilst many compounds and target hypotheses fail before the clinic, many of them actually still go on to fail in the clinic once they actually reach there. And that's a very expensive way of failing drugs. So clinical trials are very expensive with a very high attrition rate. And I think we're up to around 12%. So 12% of molecules that actually go into the clinic will fail at that point. And it's due to around things like lack of efficacy, pharmacokinetics, pharmacodynamics, so how much drug is in the system and how long it takes to clear from the body versus how effective that drug is, whether there's a wrong safety profile, or generally it's just the wrong strategy. So as we're getting sharper at looking at different and new targets, we identify them, validate, get the best chemistry, work up some preclinical data, it still feels like we're lobbing these molecules directly into the clinic without really thinking about the main questions that should be asked. It's not. I'm sorry now. Okay, so what is translational science? So applied research to support drug discovery. The really big thing is around understanding what the clinical questions are, and then building the testable and scientific evidence that allows the drug to transition between that preclinical and clinical setting. So we can generate testable scientific hypotheses, and that supports drug development. So what are the questions? So what disease are we going for? Which patients within that disease? If we understand which patients, what already is their current standard of care for their disease? Are we going in as a combination or a monotherapy? What dose level or what schedule? And it's always been around the 5 R's. So what's the target we're targeting? What's the tissue we're targeting? What's the safety of that drug? Who are we targeting within patients? And ultimately, what's the commercial potential? So there's many questions that drug discoverers have to answer before they even think about going into the clinic. So how do we do this? Understanding much more around drug and disease using biomarker information is critical. Clinicians are already in the clinic predicting, diagnosing and monitoring disease. But when a new drug comes along, they need to know much more about what's happening when they're actually prescribing this to their patients. And while disease symptoms are subjective, biomarkers provide an objective and a measurable way to actually characterize the disease. Oncology has made a lot of advances in this area, delivering really quality drugs using biomarkers. But other diseases are slower to adopt, but there's so many learnings that can be translated from oncology and into those other disease areas. So when I first started out on my big pharma job, biomarkers was not really well known. And I started on a project around an epidermal growth factor receptor, tyrosine kinase inhibitor, which was an oncology drug blocking signaling through the receptor. I joined the team just before the drug ZD1839, which its generic name was gefitinib. It was launched in Japan in 2002, and I joined the team just a year before that in the lab in sort of translational science. And it was very much, we were designing drugs where we would just put them to all comers. So we knew that lung cancer had a high proportion of the receptor in this disease, but we didn't know anything more than that. And so many clinical trials in the early days of this drug were failing because only up to 1 in 10 patients were responding. The levels of the receptor actually didn't dictate whether the patient was going to respond or not. And so it looked like the journey for this drug was going to end, and actually in a very expensive way. Simultaneously, around 2004, researchers were actually starting to sequence oncology mutations. And there was EGFR was found to have several activated mutations within its tyrosine kinase regions. Going back to some of the clinical studies that Iressa, which is gefitinib, actually had run, they sequenced some of these mutations to see whether they existed in the patients, and they actually found that patients who harbored that mutation were more likely to respond. And this was a massive leap for this drug that looked like it potentially wasn't going to go much further due to so many failings. But then there's still a lot of questions. How prevalent are these mutations? If we were to actually start from the beginning and say, well, let's look at designing a drug to these mutations, how many patients actually harbored this mutation to warrant developing a drug to it? And it was realized that actually many patients do have that mutation. And now gefitinib, or Iressa, is currently approved for treatment of first-line mutations in advanced non-small cell lung cancer patients in over 85 countries. But the story for this doesn't really stop there, because as patients are receiving this treatment, over time, a proportion of them also become then resistant, and a few of the cells within their tumor escape due to acquired resistance mechanisms. And that's now led to further third-generation EGFR tyrosine kinase inhibitors to be developed. And these ones are specific then to those sort of resistance mutations. So the T790M was a gatekeeper mutation which was identified as being a resistance marker to these types of inhibitors. And that started then a trend of new drugs being developed just to go for that targeted mutation, which meant that from the very beginning there was already a hypothesis of which patients were going to respond and which ones, and where they wanted to then take that drug into the clinic, which is a fantastic strategy because you already know where you're going. On top of that, the understanding of how to collect samples in the clinic is also quite complicated, and so you can't always keep biopsying patients in the clinics. And so new technologies identified showed that actually tumours shed DNA into the blood system. And so by isolating DNA from the plasma, clinicians could actually then identify whether patients were harbouring any of these mutations just by taking a blood sample. So this example is a bit of a phoenix from the flames, but not every drug has a happy ending that goes into the clinic and fail very expensively. So what can we do to ensure that this story doesn't happen, and what we can do to arm ourselves to make sure we develop much stronger drugs? So there are a lot of lessons learned from these types of landmark studies, and it seems a bit of a simple one, but this one is just around understanding the biology. Often we understand the target, the pathway, but it's understanding much more around what the body's doing that makes that target tick. And preclinical studies and translational analyses can really help build the hypothesis and body of data to determine whether you actually want to go down the targeted biomarker route as early as possible, because having that simultaneously along with your drug development can really improve the success. A big one for me is tissue is the issue. Collect as many samples as you possibly can. Now this can be in the clinic, but it's mainly also in your preclinical and cell models, because the more you analyze and understand exactly what's going on, the more you can build a much stronger hypotheses. If there's no sample, there's no biomarker. Pathologists are also key here, and there's often a conflict between pushing for faster studies and recruiting and getting further along the clinical development pathway versus a push for targeted health, and fast recruitment are often not the most experienced at sample collection. So it really does have to pay to see which is the way you want to go on that route. A targeted drug needs to have a diagnostic approach. So knowing what diagnostics are already out there can certainly help if there's something you can utilize without having to go down a whole new diagnostic route. So it does matter what you measure, how you measure it, but also what's quite important is how you define a positive cutoff. If you're going to be making some decisions on biomarkers in the clinic, then you need to know what's a positive and what's a negative. So building that body of evidence will help. So that all sounds quite straightforward, but there are still many, many challenges. So for all the potential impactful applications of clinical biomarkers, such as diagnostics and supporting clinical development, there's still many, many biomarkers that are just not being utilized in the clinic. So in 2016, 768,000 papers were indexed in PubMed directly related to biomarkers, and yet still so few are being entered into the clinic. So why is that? If it's so easy to write papers about them, why are we not using them? Well, it's complex and it's underestimated. The variability of a biomarker is actually very much underestimated. So by taking one small sample from a patient is just a snapshot, and actually you need to know much more around the timing of that sample being taken, the heterogeneity of the disease, and understanding the pathology. Using inappropriate samples for building the hypothesis, the discovery and the validation is often part of the problem, but also methodology limitations. Using single-plex or very low plex assays only gives you part of the story. So clinical utility considerations for biomarkers. So these are the key things to think about taking a biomarker anywhere from your sort of preclinical into the clinic. Is the underlying biology adequately characterized? Do you have enough information about what that biomarker is doing? And then how prevalent, how many patients actually have that biomarker? If it's only 1 in 1,000, it's going to be very difficult to screen and pick those patients out. But then also, how heterogeneous is your disease? Some diseases, you have to take many, many tissue samples to be able to say whether that marker is even there. So you need to understand what that prevalence and how it's distributed within the tissue. What sort of assay are you actually going to design to be able to detect this? This one's a critical one because that assay has to be reliable. It has to be reproducible. So if you actually do see some changes, you know that they're real changes and that you can then translate that and use that in the clinic. The go, no-go thresholds have to be defined. As I mentioned before, is it positive, is it negative? You need to know exactly what number you're looking at and the data to go behind that to say that that's a true answer. Study design is very important as well. You need to know when you're actually going to take your clinical sample, because if you actually take your sample too late, then you will have missed the time point in which then that marker is now no longer at the point where you needed to measure it. So the stability of the marker, if you think about circulating tumor DNA, it clears within the system in half an hour. So if your drug is hitting the target quite quickly and you're shedding DNA, if you take a blood sample to measure your circulating tumor DNA 6 hours later, you won't get your early markers. Within that, you'll get much later ones. So you need to understand the timing of the impact of the inhibitor. But then also, how many samples do you need to provide a statistically robust decision? And then samples. With all of this, biomarkers, assays, can we actually deliver all of this in a real world situation? So how can the catapult help? Well, we can help identify these key clinical questions and understand the line of sight. So researchers who have a drug, they're developing it, they've made some fantastic advances in exactly where they want to direct their drug into the clinic. We can help build that body of evidence that will translate it into the clinic. We can generate and build assays to help test that evidence and put together a package of information that will give them the most chance of success. So an example for this is, how do we know that you've got the right patients for targeted therapy? By generating disease linkage data to guide indication selection and target patient selection is key. By building that evidence and using the right platforms, you can maximize that just from very small, precious clinical samples. By impacting the impact of therapy on disease biology, by monitoring therapy, by taking samples from patients or animal studies and measuring what changes over time is quite critical to understanding the story. So the potential to screen samples from preclinical models, but also from patients who failed therapy, gives us the ability to identify novel resistance mechanisms, and all of this helps inform. So which disease, which patients? Are we going for early or late disease? Which line of therapy are we going for? Is it first line, or is it once patients have already received many lines of therapy, in which case you would need to understand what the profile of those patients look for? Is your drug going to go straight into the clinic as a monotherapy, or are you actually looking at other combination options, and what data do you need to provide to be able to do that? Is the drug you're thinking of going to be once daily? And if so, what level are you going to go in, and what's the schedule of that once daily? If it's actually going to be a short-term dosing, then safety might not need to be a concern. But if you're actually going to have a drug every day for the next 2 years, then you need to be really sharp on what your safety and tolerability is going to be. And does my drug hit my target? These are just some of the questions that you need to be thinking about before you even go into the clinic. And by understanding this data will certainly help increase success. So at the Medicines Discovery Catapult, our strategy is built around 4 interconnected focus areas. Biomarkers is one of those areas, but we feed into the drug delivery and preclinical imaging space. We look much more at building technologies around target engagement and validation to make sure that within tissue and within preclinical models, we are actually measuring exactly what we think we're looking at, and then building much more informative complex cell models. Cells just built at the bottom of a flask is just not as informative as having a much more multi-co-cultured organoid. So bringing expertise, drug discovery know-how, and state-of-the-art technologies builds into this collaborative research. We're not doing drug discovery ourselves, but we are here to help other people do it much more successfully. So how are we looking at biomarkers, and what's our approach here? So we are here to help researchers access samples through our samples and data teams. And these could be things like tissue, blood, it could be CSF, they could be urine, and we can look at a whole range of different markers within those samples through multiplexed readouts. And that allows us to understand much more about the disease, but also the drug, of the impact of the disease within preclinical and clinical samples. And that can do one of 2 things. The first one, it can understand and better give you a clinical study design. So we can develop what your proof of mechanism markers would be in the clinic, whether you're actually having a response, which would be proof of principle and proof of concept. Are there any predictive biomarkers that we can detect there that will allow us to select our patients? What safety biomarkers should we be monitoring to ensure that the drugs are safe? But what other things can we think to look for around combination options and markers of resistance? By understanding all of that collectively, we can back translate that information into our cell model group, who are building novel models that reflect much more the patient disease setting to allow drug discoverers to screen their compounds. So I'm just going to take you through a couple of the capabilities that we're building. So we have a preclinical imaging suite where we can do many different imaging techniques, and we have had a previous webinar on that, which you can access through our webpage. And just a quick overview here of a biodistribution study where we can label by zirconium and track exactly by PET imaging the distribution of those antibodies within the animal and see where they're localizing themselves. But also building a non-radioactive capability through near-infrared imaging. We can then take tissue and blood samples from those animals and actually do a whole range of biomarker analyses, including mass spectrometry imaging, where we do high content surface analysis. And that allows us to look at things like drug distribution, how much of that drug is in the tissue, but also then what's the impact on the biomarkers, and that gives us spatially relevant information. And we have a couple of technologies there where we can do mass spec imaging on. We can also take tissue and blood samples, and we can look at pathway markers. So this was some work that we did with an SME where an in vivo study was run, and they took their drug plus one other, looking at monotherapy and then in combination, and looked at a whole panel of immuno-oncology markers in combination, and just really delve into the pathway analysis of those to support a clinical trial strategy. And what we found was that the combination in this example actually showed a real high upregulation of many of the immuno-oncology markers, including things like tumor infiltrating lymphocytes. And this information is just building that body of evidence, which can give you much more information to give you the hypothesis to move forward with. In January, we'll be installing a new technology that NanoString have moved on from and building, called the GeoMx Digital Spatial Profiler. So we're participating in one of their priority sites. So I believe there's 4 of these devices being installed in the UK. And this allows doing the pathway analysis that I mentioned previously, but with spatial information. And we can multiplex up to 800 protein or RNA targets with that spatial context, which sounds absolutely fantastic, and can't wait to get this device in. And what it does is it digitally micro mirrors the tissue, and you take regions of interest, and then you can draw shapes around those regions of interest and look at the immune compartments within it. By doing that, you can then cleave all the markers, up to 800, that are within each of those regions, and then quantitatively extract and read how many proteins are within that region. And in this example that you can see on the slide, this is data from NanoString, you can see that there are hot and cold spots for immuno-oncology markers either side of that masking around the macrophage boundary. And that gives you a lot of information, especially when you have pre and post samples, so you can see what your drug is actually doing. This is another example, but using RNA. So the previous one was proteins, and this one's RNA. So they have a 96 multiplex IO target, where you can look at different regions of interest and look at the RNA expression within the tissue. And they're building these assays more and more. So more and more of these assays will become online. And what the Catapult are looking to do is actually develop much more disease and drug specific assays that we can do exactly this. And the idea with our histology group as well is to actually take the preclinical imaging, the mass spec imaging, this type of imaging, and the histology imaging, and doing the overlay of all of them, so that we can understand much more about what's happening with drug and disease. So quickly, just on to circulating biomarkers. I've talked a lot about tissue, but there's also quite a lot we can do with blood samples. So where we're getting much better at taking blood samples in patients, we can also be quite clever around what samples we can take in our animal studies, thinking about longitudinal testing so that we can understand exactly what's happening with the drug over time. So we have 2 platforms here, the Quantrix Simoa and the Biorad Digital Droplet PCR, where we can measure a range of markers at very low levels, so that if they are just being shed into the blood system at the time, we should be able to detect them. Which, you know, there's quite a lot of data out in the literature now that these things can be used in the patient, but we need to be generating and building those hypotheses in the preclinical model alongside it. So the previous one was where you already know what your hypothesis is. We also have direct infusion mass spectrometry, which is a hypothesis-free approach, where we can take just up to 200 nanoliters of whole blood with no sample preparation, and then directly infuse those samples into the mass spec and provide data-rich profiles. And that allows us to look at what's exactly happening within the patient or the animal over time. I do believe that this has the potential that we could do this also in in cell media, although there's quite a lot of salts and things that we need to think about whether we'd have to try and extract all of that first. Okay, so the biomarker discovery approach is very much not to wait till right at the end and you're thinking about going into the clinic, but the translational biomarker package should really be thought of right at the beginning of target selection. So the catapult can help with biomarker discovery, taking many, many analytes and really building that body of data right through to preclinical validation, where you can reduce the number of analytes whilst building very robust and high throughput assays ready for the clinical environment. Whilst I've been really focusing on biomarkers today, I do realise that there are lots of other areas that the catapult can help, and MDC can help navigate through that drug discovery process through our virtual R&D capabilities. So we have a team within the Medicines Discovery Catapult that can provide expertise to customers and navigate them through that pathway. And that can help them develop their target product profile. And that's really key. And part of that can be biomarkers. So we can build a biomarker strategy alongside some of these things. So you just need to come and talk to us and see how we can help with that. So just my last slide, really. I talked about the 5 R's at the beginning, but actually I think there are 5 R's within biomarkers and translational science, and where MDC can make a difference. Understanding the right clinical questions is actually key. Bridging that gap between preclinical and clinical drug development is critical. Rather than just throwing the drug into the clinic and seeing how it works, we can really identify what the clinical question is we're trying to answer with that drug, which makes us much more informed. So developing that right biomarker strategy through highly multiplex analysis, understanding the heterogeneity of the disease to identify those right biomarkers. Right assays are critical as well. We can't measure these biomarkers without knowing that the assay is robust and that we can generate that preclinical evidence that will also translate into the clinic. And the key to understanding the decision-making cutoffs. But all of this only works if we measure all of this within the right samples, and understanding the variability of the biomarkers identified is also critical. And by thinking of all of these things, we can really identify which patients will benefit. Does my drug hit the target? How long does it need to be engaged to have an effect? How prevalent is the target in the patient population, and how are we going to select them? Are we going to go monotherapy or combination, or is it even better than what's already out there? What dosing schedule and delivery, and is there a need for a resistant strategy? All of these questions we can help.